value, basically 100, and it uses an algorithm to assign individuals to a group and show similarity between groups. These two groups formed a cohesive unit 100% of the time out of the 10,000 replicates. And these results do indicate there may be a slight affinity towards Ramosissima in the Dewey Bridge population. So I calculated Goodnight and Cuellar's relatedness measure. It's just another way of assessing relationships among individuals, and this is an NMDS ordination. So each point in this ordination is an individual. Over here, the triangles are the United States population, and this is the Dewey Bridge stuff. I included some other reference samples from John Gaskin that he had identified as hybrids. So you can see the red squares clustered out over here, which are Ramosissima, and then the green circles, which is Chinensis, are over here. And most of the individuals that I sampled were somewhere in between, and there really are no clear patterns between those that were defoliated and those that were avoided by Diorhabda. Now this is a slightly different analysis called, well, I used the program Structure, which implements, it's a Bayesian clustering algorithm, which essentially can assign individuals to a group depending on number of groups that you think may be in your data set. And so I started out with, that's this K value. I said, well, I think maybe there will be 2 groups in the data set. And so the algorithm identified the Ramosissima. Clearly they were all, each line here is an individual. And so all individuals that were Ramosissima were assigned to this cluster. All individuals that were Chinensis were assigned to this cluster. And then in the Dewey Bridge population, when I said K equals 2, it had trouble distinguishing between these. Like half of the time they were put in with Chinensis, half of the time they were with Ramosissima. When I said K equals 3, you can see that it formed a whole new group. So they're clearly different from what's in Asia, but probably somewhere in between. Some individuals, maybe clustered a little bit with Chinensis, but I don't know really what's happening there. And when I said K equals 4, it did find 2 different groups between the defoliated and non-defoliated. And then I even ramped it up to K equals 9, K equals 20. It didn't matter, it still just found 4 groups. So to summarize my results, it's clear that the Tamarix at Dewey Bridge are of hybrid origin between 2 Asiatic species. There was no significant RST structure between individuals that were defoliated versus those that were avoided. However, with results from this Bayesian analysis structure, there may be slight genetic differences between the 2 groups. I think it's important to mention here, microsatellites are neutral loci, so they do not code for proteins. Selection does not directly act on these loci. They're free to evolve in the absence of selection. And so really I think where this needs to go is we need to begin looking at phenotype associated with resistance, probably leaf phytochemistry. And I'll summarize that in a bit too. But first I'm going to talk about my future research here in the Virgin River. So in October 2008, we sampled Tamarix along the Virgin River, about a mile beyond where Diorhabda reached last year. Our sample included 20, which appeared to be parviflora, 20 Ramosissima, and about 10 morphs that were intermediate. But we were really uncertain whether the Ramosissima were pure Ramosissima or some hybrid combination. And so I'll be able to assess that with these markers. I'll use more markers. And then we're going to monitor these individuals throughout the next summer just to evaluate differential herbivory by Diorhabda. You can see this is what the 2 morphotypes or morphospecies look like. And there were a whole bunch of individuals that we really couldn't identify, or they couldn't be classified into one or the other. And upstream of where we sampled, it was clear that, the parviflora showed a different pattern of herbivory than the Ramosissima. Okay, so to summarize, microsatellite data failed to detect a strong pattern of Diorhabda preference by Tamarix genotype at Dewey Bridge. But to me, this suggests relatively few genes of high adaptive significance may control resistance of Tamarix to herbivory by Diorhabda. So I think we need to include additional loci into this analysis, maybe additional microsatellites, or use AFLPs, and more reference samples at deeper levels of taxonomic divergence may help resolve what's going on. Like I said before, I think we need to characterize the phytochemistry in Tamarix in order to really know what's going on. Look at phenotype rather than just genotype. And just acknowledgements, I got to thank my daughter. She's probably my best field assistant. But also Steve Schuster, Gary Allen, and Tom Whittam at NAU, committee members, and Tom, Gail, Megan, and Levi have helped. They helped me sample in the Virgin River Gorge. Also Tony Chang has helped quite a bit in the field, and John Gaskin for sending samples. And these are just some of the other groups that I'm associated with. So I could take any questions if there's time.